Okay, for Chapter 6, we're going to be looking at two different things. The first lecture, we're going to focus on osmosis and diffusion and transport, and I've divided that into two different categories. So let me get my ink here. Okay, first we're going to focus on osmosis and diffusion, and we're actually going to do two different labs where we're going to illustrate both of those. So you may want to look at this video lecture not only for the quiz, but also for our lab. Okay, so to define osmosis, it seems a little complicated, but if you always keep in mind greater to lesser, and concentration is referring to a solution. So, for example, if you make Kool-Aid, you have a solvent, in this case would be water in our example, and the Kool-Aid itself is called the solute. So anytime you first pour Kool-Aid into a pitcher of water, you notice it's all concentrated and it has a great amount, and as diffusion occurs, the concentration from that one area gets lesser and lesser until it's mixed throughout the entire solution. So diffusion is the movement of any molecule. Any molecule is the key. Now, there is a specific type of term used when we're talking about just water, and that is called osmosis. And again, it's still greater to lesser concentration. So the direction depends on water and also the solute. And remember, we use the example of Kool-Aid for our solute. So you really need to know those two terms. Okay, so I just wrote this down again. So a solution solutes, and in most cases it's going to be sugar or water, at least for our labs. And the solvent, most of the cases, since water is universal, that is going to be the solvent. When you get to chemistry, in some situations the solvent may be an alcohol. So in this diagram here, we got a container. The blue is the water, and you notice there is a dotted line that shows a membrane. And when it's dotted like that, it shows that there are openings, and it's called selectively permeable or semi-permeable, and we'll get more to that in just a little bit. So when you actually physically, the orange dots are the solutes. It could be sugar, water, Kool-Aid. You'll notice if you count up, there are more on the right side than the left. So diffusion, since we're talking about solutes, keep that in mind, diffusion, any molecule. So the solutes in this case will be diffusing across greater to lesser concentration until everything is equal out. Okay, now here's some terms. It'll be a little confusing at the beginning, and these might be all relatively new terms for this lecture. We have something called hypertonic solution. You notice that I made all caps for hyper. Hyper refers to high. So we have a high concentration, and these solutions, we're always talking about the solutes. Okay? So hyper means high, so high solutes. And then the solvent, which is water, is always the opposite. So high solutes, low water. So that's hypertonic. Hypo means low. Sounds the same. O, low. And so again, we're talking about the solutes. So that's our focus. So low solutes, and then the water or the solvent is the opposite, so that's high. So keeping those in mind, it will help you to know what we're talking about and the direction that everything is flowing. Now this seems like a lot of information. I'm not going to read it word for word, but I'm going to pull out some key things. So here's hypertonic. Hyper, so we already said that it's more or a high solute concentration. So if I look at this diagram, here's my container. It's always very important to look at any key so you'll know. This would be an example on the test. So my blue dots is referring to my water, so very important. Okay, and they've labeled, so this would be like a cell in a container or a beaker. So if my blue dots is my water, and you notice I have more in the environment than inside the cell. So I have high water, okay, and on the outside and low on the inside. But what they've done for me also is they've indicated the percent of salt, okay? So In this case, I really need to focus on, so here is 20%, oh, sorry, here's 20% salt and 10% salt. So my solutes is going to move into the cell. And so when that happens, now in this situation, they've identified the percent of salt. So it's not the opposite in this situation. That's why I use this example. So in this case, since salt is moving in this case, water is moving out. Okay? So what happens to plant cells? What happens to the cell? So in a plant cell and an animal cell. So here's the plant cell and here is the animal cell. So in the plant cell, and we'll get to the terms vacuole in just a second, so let's just focus on what's going on. So in this case, it's going to wilt, okay? And I threw this term in here, we'll get to this in just a second. What's going to occur is plasmolysis, okay? Oops, sorry about that. Okay, animal cells, it will do the same thing. So the cell is going to shrink. So in this case, if you ever had salty popcorn, you have salt going into your cells and you get really thirsty. That's because the water's going out. And I made this little note down here also. That's why it's very dangerous to drink seawater, because there's more concentration than in your body. And so what it does is it causes you to become dehydrated. And because of that, it could lead to death. So even though you're surrounded by water, it's not a good idea to drink that. So here is going back to our key terms of hypertonic. So hypertonic, and this is how it could be worded on the test. So when the solution inside the cell is hypertonic, so key thing, cell, and then it tells me what's the movement, and I've got hyper. So, and the term relative, that just means it's comparing the two. So the cell compared to the environment, and the cell is hypertonic. I'm focusing on the water movement. Okay, so those are some terms you need to pull out of any question. Okay, so what I've done here, and if you need to do this every time, that's fine, unless you can visualize it. But I'm going to go ahead and draw it out. So my cell is hypertonic. We're assuming the environment is hypotonic because it did not indicate that, just like on the page where it indicated the 20 and the 10% salt. So I'm assuming it's the opposite unless it gives me more information. Okay, so my cell is hypertonic. Hypertonic refers to the solutes, and we said that on one of the slides earlier. So high solute, and we're assuming the opposite of water, so low water. My environment, which would be my beaker, hypotonic, low solute, because again it's referring to the solutes, and then it didn't indicate, so we're assuming that the water is the opposite. Okay, so what is the movement going to do? I remember from my definition for diffusion that it always moves from an area of greater or high concentration to low concentration. So my solutes, I can draw my arrow, so arrow high to low, and my water high to low, and that's osmosis. So let's take a look, knowing the movement. So, and this is just written out for you. Hyper, high concentration, high solutes, low water. And the question was, they wanted to know which way will the water move. And this could be one way that it's worded on the test. So in this case, water will move into the cell from the outside. And if it is too confusing drawing this box for the environment, you can just leave the box off. We'll do an activity in lab where I leave the box off. So it was wanting to know the water. So water's moving in, and if it asked on the solutes would move out. Okay, so that was hypertonic. Okay, hypotonic, again we look at all of our keys, look at any indication directions. So hypertonic solution. So my dots again is my water, and I look at my cell. Okay, in this case my cell, I've got 20% salt, 10% salt. So it looks like my salt will move out, okay, and then water will move into the cell. So if water's moving in and the focus is the water on what the cell, how the cell will react. So if I go through my definition, hypo, less or low, okay, hypo or low solutes, that means the cell will gain water. So if I think, how do my plants react in August? If I don't water my plants, okay, it's a hypertonic, it's going to wilt. Once I water my plants, then water will move into the cells and it will perk back up, okay? And again, we'll get to vacuoles in just a little bit. We'll also talk about turgor pressure. So once we get those definitions, if you want to go back and look at these slides again, these definitions just include all the terminology, okay? So that's what's going to be happen, water in, and the salt is moving out. Okay, so here's another question. When the solution inside the cell, so we're looking at cell again, is hypotonic, I want to know water. What will the water do? I set it up just like the last one so you can compare the two. So again, and this, I know this is repetitive, I've got hypo referring to my solutes. So hypo means low, so low solutes, and the water is the opposite because in this question did not indicate or give me a percent. The environment is hyper, again referring to the solutes. So hyper is high solutes, and the water is the opposite because it did not indicate anything different. So drawing my arrows, high to low, high to low. Okay, so let's take a look at how it's written out for the answer. So hypo, low solutes, high water, and I wanted to know the water movement. Okay, so just focus on answering the question unless it said what will the different parts of the solution do. So then in that case I want to know both the solutes and the water. So it just asked me about the water. In this case the water will move out of the cell. Okay, so just the opposite of the hypertonic environment. Okay, the third is called iso. Iso means the same, so it's right here. And on these longer definitions, if it helps to pause the video and then write it down, that's up to you. Okay, now you notice something different? We have an arrow with a head on both ends. That is a symbol to indicate that there is no movement. Now again, look at all my keys, look at everything before you start always. So the little dots means the water, okay? So just by looking at this, I noticed that just by counting the dots, I've got more water dots on the outside than the inside, but I need to look a little bit further. I've got 10% salt. It gives me more information on the outside or in the environment. Oh, and I also have 10% salt inside. Now since iso, hypo, and hypertonic refers to what the solutes are doing, that tells me that I kind of ignore the water and what in this situation, and what happens, there is no movement. If there's no movement of the solute or the salt, there is no movement of the water. So that's iso. So iso means the same. So the same inside and out. So I gave you a couple different examples. So high water, and again I put an arrowhead on both ends, and the solutes are the same. And I do that low, low, and high, high. You'll do a worksheet where you'll have this and you can draw that out again as practice. Okay, so that was osmosis diffusion and looking at the different types of hypo, hypertonic, and isotonic. Now we're going to move into transport, how things move. Okay, we have passive, and then in the other one we're going to have active. If anything is passive, which is forms of transport where osmosis means it does it automatically. It doesn't need any outside energy. So the example I always use would be, for example, if you're in a boat and you're just floating down the stream. I don't need to paddle. There's no extra energy involved. So no chemical energy. Okay? And that's what osmosis and diffusion does. It just gradually or automatically goes from an area of greater to lesser concentration. Like our Kool-Aid example, when I pour the Kool-Aid in and I just let it sit there and I don't use any energy to stir it up to accelerate the process, it will eventually diffuse. The Kool-Aid will diffuse. Okay, so that's passive. Active is I have to exert or use a chemical energy. And in almost all cases, the chemical energy is ATP, and we've had that before. So it needs help. So if we go back to our boat in the stream, it would be if I am paddling upstream. Like I use a lot of energy to get that boat to move upstream. Okay, so that's passive and active transport. Now, in the cell membrane, and when we get to the organelles in our next lecture, this will kind of all go together. But a channel, so this is a channel in the membrane. So, passive transport. We have these special channels, and we'll see this in our lab. I always like the example of a chain link fence. Okay, so the little holes in the chain link, now can't cheat and go over the fence, but any molecule that's too big to go through these channels, they will use a gate. Okay, and so this chain link fence is our membrane, and then we have these huge openings, our gates, that will allow the big molecules to go through. Okay, so if something's semi-permeable, what can go through our chain link fence? Well, we've got salt, okay, and water. So we know that a water molecule is very small. Salt is also very small. So the salt can go through our chain link fence. Now what's too big, and if you watch this lecture, it will help you with our lab write-up. Okay, what is too big is sugar. If I put under a microscope, a salt molecule and a sugar molecule, the sugar is much larger. And so it has to go through a gated channel. In our lab, we'll use a special type of membrane. It's called dialysis tubing. And what happens if I try to force the sugar to go through these little chains, it will actually plug up these holes. It'll get stuck, okay? And if it's all plugged up, then water or anything else cannot get through. So just kind of remember that for our lab. Okay, make a note of that. That'll help on your answering your conclusion questions. Okay, now cells also use other things to get molecules to move in and out, and it's called endocytosis, exocytosis. It sounds like a very complicated term, but anytime you see the word cyto, that equals cell. So we're talking about if we have cytolysis and cytolysis referring to the cell. Now, if I have the term endo as my prefix, that means into, to enter, okay? And exo means to exit. Okay, so if you just know how to break down the terms, you can figure out what this means. Okay, so cells are going to use these two processes. Things are going to move in and move out. Now, what it wants to do as a cell is it wants to remain balanced. So we have this special term called homeostasis. Okay, homeostasis. Everything's in balance. And they always use these two key words, biological balance. And that just means that everything is moving in and out. It's kind of an isotonic environment. So if we relate the cells, what the cells are doing to our bodies, when we're eating, we're bringing in nutrients. So that's endocytosis. So that's comparing it to the cellular level. And when you go to the restroom, that's exporting waste. And so when you're eating 3 meals a day and you're going to the restroom, your body, you feel healthy, you're good, biological balance, that's homeostasis. So at the cellular level, the same thing occurs. Okay, so let's break down endocytosis. Endocytosis, 2 examples referring to the size of the particle that's brought into, remember, endo means into the cell. Okay, so anything that's pinocytosis, that means very small. Okay, and phago is anything, I'm running out of room here, is anything that's large. So, endocytosis, so if I have pino, that's small, that would be any fluids, so water, is if it's brought into the cell, that's pinocytosis. And then for phagocytosis, we usually see this with special either cells or organisms. And I have some images here. Let me get rid of my ink so the images will show up. Okay, so down at the bottom, I probably is being cut off. This first one is phagocytosis. I'm not sure how well you can see this, but here's the cell membrane right here, and I'll kind of outline this for you. Okay, and so it is bringing in kind of caved in, and so it's going to bring in this large molecule. So this is kind of what a white blood cell will do. It's very large, and so this could be bacteria. Okay, pinocytosis is this one right here. It does the same thing, but if you look at it, they're very tiny little particles. It still concaves in, but it's the size of the particle. So there's a bunch of little ones, a group of them, and this is just one. Okay? So that's pino and phagocytosis. This is an example of another white blood cell. This blue is the white blood cell, and it actually will, this could be bacteria. So it's bringing in a large molecule. And the next one is an amoeba, which is like a single-celled organism, and it's this clear structure. Okay, it is actually eating or digesting another single-celled organism, and that's a paramecium. Okay, our next two terms, we're going to look at a term called plasmolysis and cytolysis. I know a lot of these terms look like they could be used interchangeably, but there is a slight difference. Okay, when we're talking about plasmolysis, we're talking about losing pressure. Let's not focus on the word turgor, because that's a term they use in biology when we're talking about water that is exited. Notice that it's past tense. And so when I lose pressure, if I use the example deflated balloon, let a balloon sit in my room for an extended period of time, it tends to deflate. And so it's deflated. So same thing, I lost turgor pressure. When that happens, the cell is going to wilt. So what has really happened? Now if I use the terms hypo and hypertonic again, I'm just going to focus on the water because that's what the pressure is referring to, just water. So water is moving out. So that's what's happening. Now the result will be, and this is what plasmolysis is talking about, the result, think of it kind of like a diagnosis. So the cell is shriveling up, okay, so the water's leaving. So if I go back to my popcorn example, our cells will shrivel up, and that's why you get thirsty. Now for a plant cell, okay, notice it keeps its basic shape, but inside the vacuole is going to decrease, and that's going to make it the wall not as rigid, and my plant will wilt. Okay. The next term, which is the opposite, is cytolysis. Okay, and so cytolysis is exceedingly high turgor pressure, and again focusing on water, so the cells are going to swell. This will be if I have blown up a balloon. So I'm just going to focus on the water. So in this case, the water, the movement has moved in, and so the result, the diagnosis, would be, now you notice here something has happened. In this animal cell, when I have too much water going in, it could burst. Now this doesn't happen all the time. There are special or certain diseases or conditions where this would happen. But the reason why is because of the thin membrane. So if I think of blowing up a balloon, if I keep blowing it up and putting too much air into it, it could explode. Now notice that my plant cell has not. This is a test question. Okay? The reason why is because of the cell wall. It is very rigid and very thick, and there's actually the plasma membrane on the inside, so it prevents it from exploding, basically. Now we've thrown out a lot of different terms, and so you know which term to use. You really need to read the questions carefully. So when I'm thinking of plasmolysis and cytolysis, those are conditions. So think of those as a diagnosis. So the cells will swell and animal cells would burst, or it could wilt, or the plant could wilt. When I'm talking about hypo, hyper, and isotonic, those are the directions. And you notice it always asks, which direction will the water move? Which direction will the solute move? So it's usually asking you a question. If I'm talking about turgor, that's an action. So when the pressure is low, then water leaves. When the pressure is high, okay, so that's the action of blowing up a balloon, or the action of the balloon sitting and deflating. Then here's a new term. This is called equilibrium. And this is usually once everything has been balanced and there's a biological balance, which is homeostasis, then equilibrium has been reached. Okay, so that would be isotonic. No more movement, everything's good inside and out, and so there's a balance, a state of equilibrium. Okay, I know I covered a lot of terms, and this lecture is a little bit longer. It's because I've combined two into one. So this would be one I would definitely go back and review, just because of the material is probably new to you. Okay, I will see you at the next lecture.